Before we get into how prokaryotes regulate their genes and the products that they make, I want to refresh your memory on enzymes and how they can be inhibited. So if this right here, if this right here is the active site and this is the substrate, you could see how, and this is the allosteric site, how a substrate normally fits into the active site, and then there's products made. However, sometimes there can be an inhibitor or an allosteric inhibitor that attaches to a site different than the active site, changing it so now that substrate cannot fit and there's no chemical reaction. Okay, alrighty, this is called non-competitive inhibition. So if you are one of my students making this up, it is the title of your 6 boxes is Regulation of Gene Expression in Prokaryotes. So let's go ahead and look at our intestines and zoom in. So inside of us we have lots of bacteria living, and let's look at the amino acid tryptophan. So tryptophan is an amino acid that's used by all living things. It's one of the 20 amino acids that we use to build proteins. Now humans and bacteria are very different. However, we all use the exact same genetic code. The genetic code that codes for the 20 amino acids. So for example, tryptophan is coded by the codon UAC. So in our RNA, anytime me or a bacteria have UAC, it's going to code for tryptophan. So the bacteria that are living inside of us in our intestines also need tryptophan. So they really have 2 options. If their host or the person they're living inside of eats tryptophan, well, they have access to tryptophan in their environment. But if their host is not eating too much tryptophan, then they have the option to make it themselves. So they have 2 options from getting it from their environment. For attaining tryptophan. They can get it from their environment, or they can make it themselves when their host that they're living in is not eating it. So if a bacterium needs to make tryptophan, it's going to activate a metabolic pathway. Now, as a refresher from the beginning of the year, a metabolic pathway is when you start with a molecule and then you have a series of steps controlled by enzymes that catalyze each reaction until you have a final product. Now, in this case, the final product is tryptophan. So let's go ahead and look at, in some detail, how bacteria will activate their metabolic pathways to make tryptophan. So you can see a series of genes right here. 1, 2, 3, 4. There could be 5. So we know that genes code for mRNA, and mRNA codes for a protein. And in this case, the protein it's coding for is an enzyme. Now this enzyme will catalyze a reaction. It'll take substrate A and convert it into product B. Now gene number 2 codes for a messenger RNA that codes for another enzyme. Now that chemical reaction will turn that into C. Gene 3, guess what? Another enzyme. Okay. We're finally at the end of the metabolic pathway. The last product that's made is tryptophan. So here you have a series of genes that all code for the proteins that the bacteria needs in order to make tryptophan. Now this metabolic pathway will continue. As the bacteria requires tryptophan in its life, it's going to make tryptophan and make tryptophan and make tryptophan. However, there's going to come a point where there's enough. There's enough tryptophan, and so that bacterium, it really wouldn't make sense for it to continue this metabolic pathway and make large continuous amounts of tryptophan. So in reality, there's 2 ways that this bacteria can regulate how much tryptophan it makes. So the big idea here is the bacterium is responding to its environment. In this case, we're looking at the internal environment and how much tryptophan is present. So here it's made a whole lot of tryptophan, and because there's so much, one of them will actually act as an allosteric inhibitor to the first enzyme. So a tryptophan, when the levels are so high, randomly collides with enzyme number 1, attaching to its allosteric site, working as an allosteric inhibitor, and now that first enzyme changes shape. The active site is no longer viable or no longer works. So now when you have that first substrate come in, there's no reaction. And so therefore, this metabolic pathway is shut down. Now the bacterium, though, needs the tryptophan. It's still doing protein synthesis. So that tryptophan is going to get consumed and used up in the little ribosomes as that bacteria builds the proteins it needs for life. So over time, though, the tryptophan levels are going to decrease, which also means that this tryptophan will also be used. Now, this allosteric inhibition is reversible. This enzyme number 1 will go back to its original shape, and then substrate A can attach, and you can have the metabolic pathway producing tryptophan all over again. So when we look at this process, we notice that as the tryptophan levels increased, they fed back and attached that first enzyme and inhibited it. So this process is actually called feedback inhibition. The product feeds back and inhibits that first enzyme in the metabolic pathway. But the product will only feed back and inhibit if you have a whole lot of the product. And it's a temporary shutdown of the pathway so the bacteria doesn't waste its resources continuously building tryptophan when it has sufficient amounts. Okay, so let me erase some of these lines for you. So the activity of the enzyme, of the first enzyme in the tryptophan synthesis pathway is inhibited by the pathway's end product. In this case, the end product is tryptophan. So this enzyme is going to get inhibited by tryptophan. Thus, if tryptophan accumulates in a cell, it shuts down the synthesis of more tryptophan by inhibiting enzyme activity. Such feedback inhibition, because this feeds back and inhibits, allows cells to adapt to short-term fluctuations in the supply of a substance it needs. Okay, so if you're my student, go ahead and what is feedback inhibition? You can describe that in your box number 1.